Okay, chapter 3, we'll start with concepts of cellular structure. You can skip cell theory and we go to cell shapes and sizes. As we know, organs are made of cells. The organs have different shapes. Therefore, the cells must have different shapes. And if you look at this figure right here, you can see in there that you have squamous cells, which are going to be flat cells, cuboidal cells, columnar cells. These three are very common. You have some other ones, such as stellate cells, that you're going to find in the brain, for example. You scroll down. Next concept that we need to understand is that the cells have different sizes. Well, organs have different sizes, so it makes sense to have cells with different sizes as well. So you need to understand the cells have different shapes as well as different sizes, basic components of a cell. We can look at that in this figure where you can see you have a nucleus, you have all these other structures right here. Let's take a look at a drawing of this cell. And if you scroll down, you can see here the same structure in the same cell, right? Or similar, I should say, where you have the cell right here, and this what is composed of. Then you're gonna see this green area. It's gonna be the Golgi complex. You're gonna have mitochondria right here. You're gonna have microtubules, which are these lines, a smooth endoplasmic reticulum, a rough endoplasmic reticulum, centrioles, nucleus. Okay, and we're gonna talk about those later on. Okay, the next item is cell surface. And in here we have the main topic, which is going to be the plasma membrane. You can see right here, plasma membrane right here. Okay, so let's take a look at drawing of the plasma membrane. You can see it right there. Now, we know from chemistry that we have four main organic compounds. Those are going to be carbohydrates, lipids, proteins, and nucleic acids. The nucleic acids are located in the nucleus, so they're not going to be part of the membrane. So the other three are going to be there. So you can see right here, if you look at this part, you can see the green one in here are going to be carbohydrates, the purple are going to be proteins, the golden in here are going to be phospholipids, which are fats, and the blue ones are going to be cholesterol, which are also fats. And right there you have all the three organic compounds that are located in the cell membrane. Remember, nucleic acids are in the nucleus, so you're not going to find them in the cell membrane. If we look at this again, right, we have the green carbohydrates, purple is proteins, golden is fat, in this case is phospholipids, and blue is also fat, but in this case that's cholesterol. Okay, so let's keep scrolling down, and you can see intermembrane lipids. We already covered that. Also, we said we have proteins, as you can see, intermembrane proteins. You have some examples in there. And some of the functions of these membrane proteins, as you can see right here, they're going to be receptors, enzymes, channels, cell identity markers, okay? And they also function as second messenger systems. They are called second messengers because there are going to be two messengers involved. Right there you see second messenger. Right here you have the protein. First messenger right here comes from, let's say this is a hormone. It attaches to the receptor, and the receptor, in this case a protein, is going to activate another messenger. That's why it's called a secondary messenger. If we keep scrolling down, we're going to get to the glycocalyx. The glycocalyx is a substance that is going to cover the cell membrane like an icing on a cake. Right? The cake would be the cell, the icing would be the glycocalyx. The glycocalyx is just a combination, as you can see right here, glycolipids and glycoproteins, basically carbohydrates with lipids and carbohydrates with proteins. They are very important because they are going to be unique to each individual. Okay, so each person has individual and different glycocalyx, and that's what differentiates one cell from another. Okay, so for example, my cells are different than your cells because my glycocalyx is different than your glycocalyx. Okay, now extensions of the cell membrane and the cell surface. The first thing that you have in there is a microvilli. They are called extensions because right here, more or less, is where the cell membrane is, and you can see here these are extensions. These are called microvilli. They are located in your intestines, and they increase the area of absorption for nutrients that you have at the level of the intestines. You're going to see that later on, but you need to know that the cell membrane is going to have microvilli in some cases. If you scroll down, other extensions that we're going to find are going to be cilia. They are not extensions like the ones before, look like fingers. Right? In this case, these extensions look like hairs. These extensions are going to be called cilia, and you're going to find them, for example, in your trachea. Okay, to eliminate mucus that we have in the trachea. As you can see here, these are the cilia moving the mucus that we may find in the trachea. If we scroll down, another extension from the cell membrane is going to be the flagella. Flagella in this case is located in specialized cells such as the spermatozoites. The spermatozoites are going to use the flagella for movement as well, but this is going to be for propulsion purposes in the case of the spermatozoid. Another one that we're going to find is not permanent. It's not permanent, but it happens from time to time. Are going to be the pseudopods. You have an example of this. The cell more or less goes up to here, up to this level. But you see in here the cell produces or creates these extensions that are going to elongate from the main area of the cell outside in order to collect, for example, these three substances that you see right there. So then which ones are the extensions that we have from the cell membrane? We said we have villi, we have cilia, flagella, and pseudopods. So the next topic is membrane transport. So we just finished seeing the cell membrane. Okay, what's the job of the cell membrane? Obviously, is to provide boundaries to the cell, but not only that, but also to allow cell transportation, meaning to allow things to go inside the cell and things to go outside the cell. How these processes are going to happen? Well, there are two main types of cell transportation. One of them is going to be active, and the other one is going to be passive. Passive doesn't use energy. Active uses energy in the form of ATP. What is it that passive is going to use then if it doesn't use energy? It's going to use differences in pressure or concentration. So when you have differences in pressure, for example, from high to low, if you have two areas, let's say outside the cell and inside the cell, outside the cell is 5, inside the cell is 3, then the things are going to move from the outside to the inside because it goes from high to low. Examples of passive transportation are going to be filtration. That's how things get filtered. As you can see in this figure, from the outside to the inside or from the inside to the outside, depending where you have more. Again, remember, it always moves from high to low. Now you have diffusion, same thing. It moves things from outside to the inside because of difference in the amount, right? High concentration to low concentration. As you can see right here, high concentration to low concentration. In this case, it's solutes, okay? And if we scroll down, you're going to find right here some of the factors that are going to affect diffusion. For example, molecules move faster when it's hot, so temperature affects diffusion. Molecular weight, if you have oranges on one side of the cell and you also have on the same side, let's say, an atom, then the atom will move faster through the cell membrane because it's smaller. So the size and the weight is going to be important in cell transportation. Okay, membrane surface area. If you have a cell that is 1 inch, the cell membrane is, I don't know, let's say 2 inches, and you have another one that is 2 feet, then obviously in the one that is 2 feet is going to be much easier for cell transportation to happen because you have more area. Membrane permeability. If you have a membrane made of bricks, nothing will go through. But if you have a membrane that is made of tissue paper, then a lot of things will go through because it's permeable. So obviously membrane permeability will allow things to move from one side to another. Osmosis, again, is the passage of things from the outside to the inside, but in this case, it's very specific because it relates to water. Again, it moves from high pressure to lower pressure. Again, always from high to low. So let's keep it scrolling down. And remember, as I said, always from high to low. Okay, this is an example where you need to understand. I just told you that osmosis is the movement of water, for example, but inside our body, we are not made of just pure water, right? We have solute inside. So then you cannot say that this is going to move from high to low because of the amount of water. Because in here, as you can see, the water is the same. But you cannot just consider water because you also have other molecules. So because of that, you need to consider the total amount of forces that water is going to give you and the total amount of forces that these other, for example, brown molecules are going to give you. So they're going to give you some forces and the water is going to give you some forces. Look at both containers. You have more forces right here inside A than inside B because you have in here more water. The bigger the molecule, less space for water to be located in container B. Since the molecules inside A are smaller, then you have more space to put more water. Since you put more water in here, the pressure in this side, container A, is higher than in container B, and you can see how the water will move from this container A to container B. Okay, so it moves from high to low, even though in here it appears to be the same, but it's not, because on container A you have more water. Why? Because the molecules are, these other molecules right here are smaller inside, therefore that allows you to put more water inside A, therefore moving the water to side B. So let's keep scrolling down and we can see right here, we always say high to low, right? Pressure, remember that? We always have to think about high to low, but remember, you cannot think about just high water and low water. You have to think about all the forces that you have, not just water, but also the other solids, such as, for example, glucose and salt. Okay, so how do you measure, for example, this glucose and this salt? You measure that in concentration, right? You can say that the sugar is very concentration, or the salt, and that it means osmolarity. Okay, what is osmolarity? The amount or the concentration of the solute, especially those that are not going to move from one side to the other. That's why it says in here quantity of non-permeating particles. So we said that everything moves from high to low. What gives you that? Concentration. In other words, the osmolarity. Okay. Now, what is tonicity? In this case, tonicity is the ability of a solution, okay, to affect the fluid and volume and pressure of a cell, as you can see right here. So basically what it says is that if you have the same amount of solutes inside and outside the cell, then nothing is going to happen. Because, for example, this is the red blood cell, and let's say that you have 3 pounds of salt, and this is outside the cell. This is the container where you have 3 pounds of salt. So since you have 3 pounds outside and 3 pounds inside, then nothing is going to happen to the water because it's not going to move. Therefore, this type of solution outside the cell, the type of solution, is going to be called isotonic. If you look at this other case right here, this is a hypertonic solution. Using the same example, this is going to have inside 3 pounds of salt. Outside, you're going to have 5 pounds of salt. Because of that, the water will go from higher here inside the cell to outside the cell. And that's why the cell will shrivel. Right here, this is hypotonic. In hypotonic, you have in the cell 3 pounds of salt, and outside you're going to have, let's say, 1 pound of salt. You have more salt in here, therefore the water will go from the outside to the inside, and therefore the cell will swell and eventually may explode.